Hello, my name is Alex Jahn, and I'm a biologist at the Environmental Resilience Institute at Indiana University, Bloomington. My specialty is animal ecology and behavior, and one of the projects I'm pursuing right now is on American robins and disease, which is what I'll be talking about with you today in this brief talk. Now, birds are all around us, from forests to backyards and city parks and farms across our country and the planet. And these birds are playing a vital role within these ecosystems that we share with them. For example, they control insect populations. Birds such as the swift at the top left of the screen consume numerous insects in any given day because they're constantly flying and eating. Same goes for whippoorwills and nightjars that do so at night. Other birds, such as this eastern kingbird in the lower right-hand side of the screen, eat fruit and disperse seeds, and they effectively plant our forests that way, preventing erosion and providing forest cover for other organisms. Other birds, such as this ruby-throated hummingbird at the top left, pollinate our flowers, and birds such as this turkey vulture at the lower right-hand side of the screen are scavenging. Basically, vultures provide that kind of service in terms of cleaning up carcasses in the environment. And not only do birds provide these types of so-called ecosystem services, they also just provide a richer experience for us as humans when we go out in nature. Can you imagine going out and not seeing any birds, even on your way to the store? These birds are pretty much everywhere, and we might take them for granted sometimes, but I'm sure we'd notice if they weren't around. So birds really provide a lot of benefits to us as humans in many ways. And they also are harbingers of disease, which I'll be talking about in this talk. So tick-borne diseases in the United States are probably more common than you think. So these are examples of 6 tick-borne diseases that affect humans in the United States. Each map shows the locations in the country where most human cases of infection have been reported. And if you look at the map for Lyme disease there in the lower center of the screen, you can see it's most common in the northern Midwestern states and the northeast of the country. And this is the disease I'm going to be talking about for the rest of this talk. So Lyme disease is the most common vector-borne disease in the United States, and it's caused by this bacterium Borrelia burgdorferi. So a vector-borne disease is a disease that's transmitted from one host to another through a vector. And a host could be, for example, a deer that is infected with Borrelia, this bacteria that causes Lyme disease. And the vector would be the tick. After the tick bites the deer, then it might go ahead and bite another deer, infecting that other deer. So the deer serve as the hosts, and the tick is serving as the vector. Disease cases in Lyme disease across the United States are present throughout most of the country, but most common in the northern Midwestern states and the Northeast, as you saw in the previous map. And this map here shows the percentage of cases across these different states. So it's a disease of concern for the country because there's no vaccine to prevent people from getting it. And as you can see from these maps, it's quite common. And it's also increasing in prevalence in the human population in the United States. This graph shows the increase since 1991 in the number of reported cases in humans of Lyme disease. So it's important to understand the life cycle of this disease and the potential risks to people living in different locations so as to prevent further infections by this disease. Now, interestingly enough, it turns out that some birds, such as American robins, can act as reservoirs for Lyme disease. And these two articles here I'm showing on the screen have found evidence for the fact that not only deer and mice that are out in the forest could harbor Lyme disease, this Borrelia bacteria that causes Lyme disease, but also American robins. So let's take a quick look at the life cycle of ticks in this disease. So if you focus first on that adult tick in the lower center of the screen, it will be looking for a host, so it could latch on to a deer or a raccoon, for example. And with the blood it gets from that host, it'll lay eggs out there in the environment, and those eggs will hatch and will latch on to a new host, maybe a squirrel or a robin, as you can see there in the top of the screen. And with the blood it gets from those hosts, it'll become a nymph. And that nymphal tick will, again, go out into the environment, maybe hang out on a branch or a leaf. And as robins are foraging in the leaf litter, or squirrels also, those nymphal ticks will latch onto that new host. And if those nymphal ticks have become infected by their previous host, by latching onto, say, a squirrel when they were larvae, and becoming infected with this Borrelia bacteria, it can transmit the Borrelia bacteria to a new host. And not only could it latch onto a robin or a squirrel, it can latch onto us when we're out there enjoying nature. So that's why it's important to do tick checks when we get back home after being in the woods. So this is the basic life cycle of these ticks, and therefore of this bacteria, Borrelia, that they're carrying. Now, these ticks might latch onto a mouse, such as this one here in the left side of the screen. You can see several ticks on its ears there, and infect that mouse. And these mice are quite abundant in some habitats, such that they provide really good hosts in terms of abundance. A lot of opportunities for this Borrelia bacteria to exist in any given habitat where they live, where these mice live. However, these mice aren't moving very far. They have small home ranges, or basically the area that they spend their lives in. Whereas for birds, although they might get fewer ticks than, say, a mouse or a deer, they will fly, and some may even migrate. Some may even migrate hundreds of miles. And so therefore, although birds might carry fewer ticks, they might be able to disperse this bacteria around much more effectively than a mammal could. In the case of American robins, it turns out that they can infect ticks that latch onto them. So once a robin is infected with Borrelia, it can infect new ticks that bite that robin, therefore passing on the disease to these other ticks with a pretty high success rate of 90%. So it seems that robins are quite good reservoirs for this bacteria. And you can see in the lower left-hand side of the screen, this robin has picked up a tick there on its face. It probably picked it up when it's foraging down in the leaf litter, and I'll be talking a little bit more about how we search for those ticks here with our research project. So just a quick overview of American robins. A lot of you probably know them if you live in the United States or Canada. They're common backyard birds across North America. They're found primarily in the summer or the breeding season when the robins have nests up in Canada that time of year, but they're found in most of the lower 48 states all year round, and only in winter in some locations in the southern part of the continent, such as Mexico and southern Arizona and southern California and most of Florida. So they are migratory in some parts of the range and probably not so migratory in others. And if we look at eBird data, eBird, for those of you who are birders, you've probably heard or used this app where bird watchers can upload their observations onto this app and therefore share their data with other people. And this map here is created using that eBird data. So what the researchers that create eBird have done is modeled using the data from birders uploading their data over time, the movements of robins. So you can see how the map is changing in colors, and that represents where most robins are located at any given time of year. So over there on the right-hand side of the screen, you can see the week of the year as this timeline moves across the year from winter into summer. The population of robins starts going northward into Canada and Alaska, and then southward again in the fall. And the darker colors represent the higher abundances of robins. Basically, people are seeing more robins in those locations, and then the lighter colors are representative of lower densities of robins. So you can see here that there is a pretty obvious signal of movement in the American robin across the continent. However, this doesn't tell us what individual robins are doing. For example, what are robins in Indiana specifically doing? Are those abundances that are moving through the state representative of birds that are spending the summer here and breeding and then moving? Or does it represent a mixture, a combination of birds that are maybe breeding up in Canada and then spending the winter in Indiana, and then maybe others that breed here in Indiana are moving as well? But we really don't know exactly how many robins of any given population, say of Indiana, are moving or staying. And that's what is important to know when you're talking about disease ecology, because individual robins are carrying these diseases. We need to have a much more clearer picture of individual level movements and not just what the species as a whole is doing. And that's what one of our objectives in this project is. So yeah, the questions that we're trying to answer is, what is the annual cycle of robins in Indiana? Where do individual robins go to spend the winter, for example? Second of all, how do urbanization and robin migration interact here in Indiana to potentially drive the transmission cycle of this tick-borne disease, specifically Lyme disease? And finally, how are these patterns changing over time with increasing urbanization and climate change? And my collaborators on this project are Ellen Ketterson, a professor in the Department of Biology at Indiana University, and she's also a member of the Environmental Resilience Institute, as is Dan Becker. He's a biologist in the Department of Biology at Indiana University and also a part of the Environmental Resilience Institute. Ellen's specialty is animal physiology and ecology. She studies primarily birds, and Dan Becker's specialty is disease ecology. So what we're doing is catching robins right here in Bloomington, Indiana, throughout most of the year. You can see, for example, this female robin on the left-hand side of the screen was caught right there on campus at Indiana University, and these two other robins here and on the right-hand side of the screen were captured. Now, we're capturing these birds in mist nets, which you can see in the figure. They're special nets that ornithologists use to catch songbirds. They're practically invisible. And what we're doing is trying to set up these mist nets in different parts of Bloomington to capture birds in different habitats. So you can see this robin on the left-hand side of the screen was caught in a more urban environment on campus, and the two males on the right-hand side of the screen in a more wooded environment. And that'll give us an idea of how different robins are potentially at greater risk or at lower risk of getting infected by this bacteria, Borrelia, depending on the habitat that they inhabit. And once we catch a robin, we're attaching a numbered band to it. So you can see the band there on the leg that I'm attaching to this bird. It has a unique number combination such that it's going to be the only robin with that number in the United States. It's a unique number combination for that one robin. And therefore, if anybody else catches that robin in the future, or even if, especially if we catch it again, which is most likely, then we'll know which bird, when that bird was originally banded, what its health was at the time of banding, and then we can track its health over time that way. And after we attach that numbered band, we also attach some colored bands, which will identify that individual with a specific color combination. And that allows us to identify that robin without having to catch it again. We can just look at it through binoculars and say, oh, yeah, that robin has a blue band, an orange and white band, and therefore we know it's that specific individual we had banded on a given date and location. And if you're birding in Bloomington, you might see some of these robins flying around because they're moving across Bloomington, as I'll show you in a little bit. And if you see any color banded robins, feel free to send me an email. I'd be very interested in those observations. They'll even help us with our research, and I'll give you my email here at the end of our talk. And now, after taking several measurements on these robins, we'll also attach a GPS tag to some of them. As you can see on the robin in the left-hand side of the screen, I'm putting a GPS tag on its back, and that GPS tag will give us its position every couple of days throughout the entire year. We have to recover that GPS tag back because it doesn't transmit data back up to the satellite. It'll just collect data from the satellite and store it on board the tag. So the way we know how to get those tags back is because we're putting those tags only on robins that we know are actively breeding. And what robins usually do is come back to the same territory year after year to breed. So we know where to go back next year and catch it again to recover that tag and download its data. And on other robins, we're collecting what's called an accelerometer. As you can see in the robin on the right-hand side of the screen, these accelerometers are made right here at Indiana University, Bloomington by a professor called Jeffrey Brown and a student of his, Chen, who are engineers in the department here of engineering. And they're making these accelerometers for us to be able to understand the activity patterns of these robins throughout the year. So that accelerometer won't give us a position data like the GPS tag will, but it will give us how active the robins are. What time of day are they foraging? What time of day are they waking up and singing, and what time of day are they sleeping? So that kind of activity level data will also allow us to understand the times of day they might be more at risk of picking up ticks. But right before releasing the birds, we will collect some blood from them. And this blood, you can see I'm holding a drop of blood in the left-hand side of the screen in a glass cuvette, which we'll insert into an analyzer to measure such things as the hemoglobin of the bird, which tells us something about how healthy that bird is. And we'll take a drop of blood also and smear it across one of these glass slides on the right-hand side of the screen, which have been tainted with a purple dye. That tainted blood that is now turned purple, we can view it under a microscope and count the number of blood cells that the bird has, red blood cells, white blood cells, and that also gives us an idea of its health and immune condition, how well its immune system is working. And you can even see some diseases under the microscope that way, such as protozoa that cause avian malaria. Now, what is the annual cycle of robins that live in Indiana? That's the first question we're trying to answer. And the way we're collecting this kind of data, again, is with those GPS tags I showed you on that robin in the picture. And this is a picture of the city of Bloomington and outskirts, with each green dot or red dot representing the location of a robin during different times of the year. So all these dots represent together about 8 robins that we've collected GPS tags from. And you can see the robins are moving across Bloomington, they're moving across campus, and they're even moving into the woods just north of town. And so it's interesting because it looks like they're using a lot of different types of habitats during different times of the year. And here's the example of the movements of just one of those robins. So it's moving again across some more urban sites in town and across campus. You can see campus there in the north part of the image. So the top center is you can see the football stadium there on campus, and the robin has been moving around that part of campus as well as out into the woods just north of campus. So with this kind of data, what we can do is combine the data we collected from it when we had it in our hands, when we captured it, the health data, its health status, whether it was sick or not with Borrelia, and then combine it with this movement data to understand whether that kind of movements between, say, the forest and the city increased or decreased its chances of becoming infected. Now, some of these robins in Bloomington are even migrating. And here are examples of 2 of them. A female on the left, number 2530, she decided to go down to southern Alabama to spend the winter, where she even moved across part of Alabama during the winter. She didn't stay very still. And that's interesting because that could affect her chances of becoming infected, maybe even down in Alabama with a disease. And she made a few stops even there in Tennessee, you can see along the way. And another bird, this male on the right-hand side of the screen, number 2532, he went to northern Florida to spend the winter. Made a few stops there, it looks like, in Tennessee and North Carolina. So these movements of the birds are also very informative for us because we might be able to compare how different birds migrate, and therefore their chances of becoming less or more infected with a disease depending on how they migrate. Now, what I didn't show you from these figures is that, well, let me first show you that there's some data here that we have from 8 birds showing that they're migrating across different states. So again, there's that one male that went to Florida. And then other birds have gone even further west, like one bird went to Texas. You can see there in the lower left-hand portion of the screen, that bird went to Texas, another went to Arkansas, one went to Mississippi. So these robins that are migrating are also moving into different wintering locations, which might increase or decrease their chances of becoming infected during the winter, depending on where they go. And what I didn't show you in these figures is that 2 of the robins actually didn't migrate at all. They spent the entire year in Bloomington, which is interesting as well. Now, that's called partial migration, where some birds of a population migrate and others don't. And why some robins might migrate and others do not is still a mystery. We're going to be studying that as well. And this data from Bloomington on robin migration is interesting because we found previously that robins in other parts of the country are migratory as well. At least some of them are. You can see we caught robins up in Denali National Park, shown in the top left-hand side of the screen, and they primarily went to the Great Plains as far south as Texas to spend the winter. We caught another one in Amherst, Massachusetts, which went to South Carolina. You can see there on the right-hand side of the screen, it went to South Carolina to spend the winter, and a couple others that we caught in Washington, D.C., they just spent the entire year there in the neighborhood of Georgetown. So across the country, it seems like there is different levels of migration. So again, robins, depending on where they're breeding, where their population is located, may or may not migrate. And the second question, again, how we want to answer is, how does urbanization and migration act in concert to affect the transmission cycle of these tick-borne diseases? It's a question we're still pursuing. We're just begun collecting data, so we really can't answer that just yet. But the way we're doing it is by sampling different parts of Bloomington, sampling robins in these different parts, basically going out there with those mist nets and catching them. So yeah, with this kind of scheme where each white circle represents the type of location where we want to catch robins, we can answer these more detailed questions, such as where do specific migrants or resident robins go to spend different times a year, and are robins on suburban or campus sites more or less likely to become infectious than, say, robins outside of town. And again, this map of how robins are using different parts of Bloomington will inform us of those types of questions, will provide information to address those types of questions. And finally, how are these patterns changing with urbanization and climate change? This is another question where we don't have data to answer just yet. We are going to be answering it hopefully very soon with this type of data we're collecting. But the take-home message here is that with that data that we're collecting from robins on collecting their blood to analyze their presence or absence of Borrelia and their movements, we can understand the annual cycle of these backyard birds and other wildlife for that matter, and therefore better understand not only their basic biology and ecology and behavior, but how to improve human health in Indiana and other places with that kind of data. And it also gives us an opportunity to just provide a better experience for people to appreciate nature, because this kind of data that we're going to produce on how robins are using different environments will hopefully provide inspiration for future generations of scientists to study birds and other wildlife, as well as disease. And I leave you with a short quote from Rachel Carson, who said, "We spray our elms and the following springs are silent of robin song, because the poison traveled step by step through the now familiar elm, leaf, earthworm, and robin cycle." And when she was making the statement several decades ago, this cycle was probably very poorly known in terms of how the robins are interacting with their ecosystem through their diet. And a lot of that is still true today. I mean, we know a lot more about robins maybe today than they did in Rachel Carson's time, but there's still a lot we don't know about familiar backyard birds like the robin. Amazingly enough, we're just starting to find out right now where robins go to spend the winter, one of the most common backyard birds in the United States. And this is true for many other backyard birds. And we need to get this information not only in order to better understand the wildlife that share our space and ecosystems with, but to better understand how they interact with their environment, such as bacteria that cause disease and which could infect us. And therefore, it gives us a chance to improve the quality of life for future Indiana and residents, as well as people in many other states. And feel free to email me to that email address there at the top left-hand side of the screen with any comments or questions or suggestions. More than happy to receive your emails, as well as your observations of any color-banded robins you might see out there. I'd like to thank my collaborators here at Indiana University and other institutions, as well as the Environmental Resilience Institute, for all their support of this project. Thank you for your time.